check.
Hello, everyone, and welcome to the Neural Implant Podcast. Today's guest is Dr. Warren Grill. And if you know anything about neurostimulation, neurotechnology, then you probably know this name because he is a mogul in this field, and he is very prolific. He's published something like 250 peer-reviewed papers and nine issued patents, 5 pending patents. Just he's everywhere. He's at Duke University, biomedical engineering professor over there, and then he's of course involved in lots of companies, spin-offs, everything like this. So this is one of the people that has been a long time coming to come on the show. I can't believe it's taken so long. And we had talked previously, but it's just one of those things. I don't know. It's really weird, but hopefully you guys enjoy it. Okay. Okay, Warren Grill, pleasure to have you on the show. You're one of the people that is just, I can't believe it's taken this long to have you come on because, just so it's like such a pivotal person in the field of neurotech, neural implants. And, just somehow, I don't know. And you're definitely one of the people that, we've talked in the past and some guests, I meet for the first time as we're talking, and then we met. I remember a few years ago at SFN, and other times too. But yeah. You are a biomedical engineering professor at Duke University. You are chief scientific officer at NDI Medical and scientific advisor SPR Therapeutics, and you've published something like 90 papers in the field of neurostimulation. Is that accurate? Is that number still accurate?
Well, the total number of peer-reviewed journal publications that we've published is about 250, and that's across a range of topics from kind of fundamental elements of neural stimulation all the way through clinical feasibility studies in human.
Okay. Yeah, so I introduced you a little bit. Do you want to talk a little bit about your work and how you've managed to publish 250 papers?
Well, I think we manage that by having an exceptional team of people. That's really, I think, the key to success in any endeavor, or the people that you're able to gather together to help realize your dreams. And I've been really fortunate for my career to be surrounded by great people. First, when I was a student, by great mentors, and then once I was running my own lab, being able to recruit fantastic students and postdoctoral trainees and professional staff to work with us. We're broadly interested in using electrical stimulation of the nervous system for restoration of function. And as I intimated when we talked about publications, we really work from trying to understand very fundamental questions about how electric fields interact with neurons and how we can control that, through preclinical studies in animals, all the way through clinical feasibility studies in human. And then that last step we've also gotten involved in, which is how do you get those innovations out of a university lab to actually impact people? And that's really the commercialization side of things. And we've done that both by patenting our innovations, that then some of those patents have been licensed and other people are now using that technology. And in a few cases, we've started companies because we felt like that might be the best way to get that innovation out into clinical use. And the theme that permeates our work is this little moniker that I'll use sometimes on my closing slide is, from understanding comes innovation. So ultimately, we want to come up with innovative therapeutic approaches to treat neurological disorders, diseases, and injuries. And in order to do that, we need to understand the pathophysiology of the system, what's gone wrong in those conditions, and how does electrical stimulation that you apply interact with that system that you might be able to right the ship. And I think without that understanding, it's pretty hard to do innovation. If you try enough things, you might get lucky, but the approach that we take is much more of a mechanistic driven one.
Yeah, fascinating. I'm curious because what What is it that you're doing, I guess, that maybe others are not doing, that in this route to understanding? I mean, I guess you're above average in this case, and so what is it? What do you think that you're doing that the others are missing, and what's the secret sauce, I guess?
I've never thought about it that way before. I don't know if there is a secret sauce. I think a couple things that are true of the way we approach these problems. One is that I learned from my mentor Tom Mortimer is trying to break things down into very simple principles that we can understand. And if you can't do that, you probably don't understand well either the question or the data or the system that you're studying if you can't really distill it down to some relatively simple principles. So I was looking at some data today with a postdoc in our group, Ati Gavazi, and she's looking at measuring the electrical properties of the tissues that constitute a peripheral nerve. Because we build models of these peripheral nerves, and it turns out that the parameters, the electrical conductivity that we use for those tissues, is really poor. Those parameters are very poorly defined. And we were looking at some data, and I said, well, to me, this kind of looks like two resistors. These are very complex simulations she had done, but I tried to distill it down to two resistors in parallel, and how the data should behave based on that very, very simple model. And in fact, in this instance, the data didn't really make sense from the perspective of that model. So she's going back to dig a little bit deeper and try to understand why things look the way they do. So one is, try to distill it down to very simple principles. The other is, I think, patience. So we have ultimately a therapeutic goal in mind, but don't rush toward that therapeutic goal. And the reason I think that's so important, and to me, this is of broad importance to our field, is that if we go too early to human, for example, and we fail, that approach might forever be tainted. Even though it could have enormous promise, people will say, oh, we've tried that and we know it doesn't work. And to me, that's really an unfortunate conclusion for people to draw from early human feasibility when it sometimes doesn't work, yet the fundamental idea may have enormous promise, but is forever or for a long time tainted. And so we really try to be patient and build up to that clinical feasibility study so that we have a pretty good idea of how things are going to work in human when we get there. And as I opened with, I think, again, people. Being able to recruit and retain really outstanding people and giving them, I think, by most standards, a pretty long leash. So with our doctoral students, for example, when they come into the lab, we'll have a pretty well-defined project for them to start on the first 3 to 6 months that they're there in the area that they think they might be interested in pursuing their doctoral work in. But the goal is to have them develop their doctoral thesis organically from their ideas in the context of the other work that's going on in the lab. And we've had multiple examples of people coming up with really great ideas that were not part of things that were going on in the lab and turned out to really be footholds into entirely new directions for research. And so I think empowering those people to pursue their passions and their ideas is really important to our overall success.
Do you think that's more you found good people? I mean, yeah, a good name, attracts good people, maybe the best or something like that. Or are you unleashing something that's within maybe everybody?
I think I think we do get outstanding people, but I think we also unleash their capability. And I think part of what makes this work well is the generosity of other people in our lab. So people come in from different backgrounds and with different expertise and different life experience, and they see the world differently, and we all benefit from those different points of view. But they may need assistance, training, a tool, whether it's surgical or statistical or how to use the microscope, right? These things that you just don't know innately. And we really have a community and a culture of generosity where people are willing to put down their own work and walk over to the other side of the lab and help you figure out your problem. And then, of course, when somebody needs your help, you've been inculcated into this kind of behavior. And so you're willing to put down your work and go help them. So once we built this and got that ball rolling, it's been self-sustaining. And I think that kind of supportive environment really allows people to be a little bolder, gives them confidence to try things that they might otherwise be unwilling to try in a less supportive environment. And so I think that's a real part of the key to getting people to blossom intellectually.
Okay. Yeah, that's really cool. I guess especially in the beginning, you're probably going to be asking for a lot of favors, so you build up a big debt, and so later you have to pay it forward. I guess that's a good way to get people to help each other out. And I might have missed it. How many people did you say was in your lab? Postdocs, PhD students, blah blah blah.
So not including undergraduates, we have 25 people in the lab, and that's a mix of doctoral students, postdocs, and professional staff. And then any given time, we probably have half a dozen undergraduates. And the undergraduates are being mentored not by me, but by one of the PhD students or one of the postdocs. And it took us a while to figure out that kind of arrangement, but it works remarkably well for two reasons. One, it allows that graduate student or postdoc, or actually in a couple of instances, staff members, to amplify their own effort, right? They can have somebody else doing something and they don't have to do it now, so they can spend that time doing something else. But perhaps more importantly, it gives them the opportunity to mentor and to lead. And they're going to be expected later in their career to mentor and help and train people. And for better or worse, we don't really get um, any training in how to do those things. It's really, I was never trained how to mentor. Remarkably, I was never trained how to teach either. And so I feel like them having the opportunity to mentor undergraduates gives them the the ability to practice mentoring, to practice working with someone else, and that's going to benefit them later in their career.
Yeah, definitely. It kind of exposes you to something, and then you can see like, do I like it? Do I not like it? And potentially grow off of that, or continue with that, or not continue with that if you realize you don't like it. Yeah, so maybe let's get into the science of things. You've been working on electrical stimulation, design analysis of electrodes. What is I guess what is the thing that you believe that's maybe a little bit controversial in the field?
Hmm. Well, I think something that uh, that's kind of timely, maybe given that today was the Bioelectronic Medicine Research Summit hosted by Neurotech Report, right? So, is this the multitude of devices that are doing ostensibly transcutaneous stimulation of the vagus nerve? And several of these devices have clinical data to support their efficacy, and they have regulatory approval for marketing and sales in the United States. And they have, as far as we can tell, very benign risk profiles. So I'm not saying that those devices shouldn't be used for the purposes for which they've been tested, but I think it's highly unlikely that they are stimulating the vagus nerve. That is, if you put an electrode on the side of someone's neck and you turn up the intensity to a level where you would stimulate axons in the vagus nerve, in the cervical trunk of the vagus nerve, The first axons you would stimulate are those that go to the skeletal muscle of the airways and throat. And that's why, if you look at vagus nerve stimulation for epilepsy, for example, the preponderance of side effect is impact on speech, hoarseness, coughing. That's what limits how far up you can turn the knob. Yet with these transcutaneous devices, there's no evidence that they're activating those most excitable axons in the cervical vagus nerve that would lead to contraction of the skeletal muscle of the airway. So I think it's probably more likely that rather than stimulating the cervical vagus nerve, they're stimulating some other branch of some other cranial nerve, and that stimulating that nerve is mediating the clinical effects, because as I said, they do have clinical data to show efficacy. And I think this is important, right? Some people might say, well, who cares? I mean, it works, so why do we care? And I think it's actually really quite important, because if in fact they're stimulating a branch of some other cervical vagus nerve, then you could probably design a device to do that really well, instead of thinking that you're stimulating the cervical vagus nerve and, oh, just really what's happening is you're stimulating this other branch that happens to be running nearby, even though that's not your intended target. So whether that's controversial or not, it's just something that I think is timely, and it's been on my mind, and I think it's highly relevant to our field moving forward in an informed way, rather than a way where we just kind of try things and see what happens.
No, I think that's very controversial. That might be one of the most controversial things I've heard on the show, actually. That we may be wrong in the mechanism of what's actually going on. That's really cool. I mean, I'm sure you could cross-validate that with like an implanted vagus nerve stimulator, and if it has the same effects and same constriction of throat or speech or something like that, then you could cross-validate that as well. But yeah, that's really interesting. I wonder if that's going to open up a dialogue for this kind of stuff. Okay, maybe we can move to less controversial stuff. What is some kind of takeaways that you've had in your career about stimulation, restoration of basically abilities for the body, basically prosthetic control, right?
One of the things that has been a little surprising to me is, well, first let me say that we work in a number of systems, right? So we look at peripheral nerve stimulation for restoration of bladder function, deep brain stimulation for treatment of movement disorders, spinal cord stimulation for treatment of chronic pain, vagus nerve stimulation, almost from the point of being application agnostic, but just trying to develop approaches to allow stimulation and block of targeted nerve fibers without block or stimulation of untargeted nerve fibers. And then in collaboration with some colleagues here at Duke, I've even gotten involved in transcranial magnetic stimulation, a totally non-invasive way to stimulate the brain. And people have said, well, that's crazy. How can you be doing all of these different things? And something that's been surprising to me is the degree of crossover. So that is, we learn something in one of those systems and realize that we can apply those same principles in one of the other systems. So I'll give you a concrete example of that. We started manipulating the temporal pattern of deep brain stimulation, that is the timing between the pulses, first as a tool to try to understand mechanism of action, and then as a potential way to improve the therapy. So we had a hypothesis that emerged from computer modeling that regularization of the firing of neurons, that is pacing them at 130 hertz, which is the frequency that most deep brain stimulation is delivered, is important to efficacy. And then if you weren't able to cause those neurons to fire regularly, the efficacy of the therapy would be not as good. And we came up with the idea of, well, what if we stimulate with random patterns? That is, the interpulse interval is random rather than being fixed. the hypothesis would say that the more random you made the pattern, the less efficacy you should observe. So at the time, you couldn't make such a pattern with an implanted pulse generator that was used for deep brain stimulation. So we first had to develop an intraoperative approach where we made temporary direct connection to the implanted DBS lead while the surgeons were changing the batteries. So these devices historically were exclusively powered by primary cell batteries. They last 4 or 5 years. The patient comes in, they make a small incision on the chest to take out the old pulse generator and put in a new one. And during that time, we would, with IRB approval, of course, and participant agreement, can make temporary direct connection to their lead so that we could deliver these random patterns of DBS. And the patients are awake because they can do that battery change under local anesthesia. So we assessed both tremor in a group of individuals who have essential tremor and bradykinesia in a group of individuals with Parkinson's disease and delivered random patterns of stimulation. And what we observed was consistent with our hypothesis. The more random the stimulation, the less effective it was at treating symptom. And that was satisfying. But more importantly, it hit us, it gave us the idea of, well, if pattern matters, what if we could design patterns that actually work better than conventional 130 hertz non-random DBS? And by better, we thought about 2 dimensions. One is efficiency, right? If the batteries only last 4 years, could we make a pattern that's more efficient, requires fewer pulses per unit time, and therefore would allow the stimulator to last longer? Or could we make it more effective? Use the same amount of energy, but do a better job at relieving symptoms. But if you think about the design space for temporal patterns of pulses, kind of think about Morse code, there's a lot of different ways you could arrange dots and dashes in different patterns. So we first had to develop a whole engineering design approach that allowed us to design these patterns. We then did that design process and used this same intraoperative approach to test those patterns in humans and showed that, in fact, we could identify patterns that treated the symptoms of Parkinson's disease more efficiently, using about 1/3 of the energy of conventional high-frequency DBS, more than double the battery life of the implantable pulse generator, or more effectively, do a better job at treating bradykinesia or slowness of movement. So that was the evolution of this idea of temporal pattern as kind of a new dimension of stimulation parameter, right? Before this, stimulators didn't have a knob on them that said pattern. But then we ported that idea over to spinal cord stimulation for chronic pain, because we recognized that in that system, because similar to DBS, you're engaging a circuit, an interconnected circuit of neurons, we also could potentially, by manipulating the temporal pattern of stimulation, improve the efficacy of spinal cord stimulation for chronic pain. And that work has proceeded through first computer-based design using the same kinds of approaches we had developed in DBS, but now with a model of spinal cord stimulation. Then preclinical studies in animals. And then the next step will be translational studies in humans who have pain to see if what we've predicted in computers and animals actually works in human. So what I think to get way back to your question, what it illustrates is this idea that a concept or a theme in one domain, in this case deep brain stimulation, can be carried over potentially and applied in what people might think of as a completely orthogonal kind of domain, in this case, spinal cord stimulation.
Yeah, that's fascinating too, and I actually think that that's a big advantage too, that you have a big group that goes into so many different directions, because then when you do find something like that out in DBS, you can those people, those postdocs, those PhD students, undergrads even, are working together in the same office area, and they're spending hours per week together, and I mean, of course, you're publishing the data, and anybody theoretically can learn about this. this, but they're going to spend hours total, working on this versus hundreds of hours working with that person and really changing the way that they think. So it's a much more efficient way of transmitting knowledge, I guess. So I basically what I'm trying to say is like everybody in the neurotech community should come work in your lab at the same time.
Well, I'm not sure about it all at the same time, but we frequently do have visitors, and we welcome anybody to come by and spend some time with us. Um, it's interesting. We had talked before we started recording about the consequences, the impacts of COVID, and one of the impacts for our group has been on kind of interpersonal creativity. And what I mean by that is these students are in the same space for many, many hours a day, many days a year under non-COVID conditions. And so someone will have some image up on their computer and somebody will come over and start talking to them about it. And then another person will glom on, and suddenly you have this little nucleus forming. And those people will be talking amongst one another. And out of those interactions often come really great ideas, right? Because people are seeing the same thing in different ways. They're seeing a new thing for the first time. They're bringing different perspectives to it. And that has been in my view, one of the biggest negative consequences of COVID are the lack of those kinds of interactions. And we've tried a couple of online meetup things, but it just hasn't worked like those kinds of things have worked in the physical space. And so I really look forward to people getting back into that shared physical space, because I feel like we're going to have a little period here where there are a downturn in new and interesting creative ideas just because of that lack of spontaneous interaction.
Yeah, definitely. And I mean, a few minutes here and there, a few times a week are really additive and can, make a huge difference. And then speaking of collaborations, I heard that you and Doug Weber are going to have a collaboration. So what hot and juicy tidbits will be coming off the Weber grill this summer?
I'm not aware, so that's news to me.
Oh, sorry. The
I'm
joke
not
was
aware of
Weber
any.
grill.
Yeah, I get it. I get it.
No, no.
But
Yeah.
I won't necessarily acknowledge it, but I do get it.
Sorry, bad jokes. Bad jokes are allowed on the show.
Yeah, it's fine.
No, but yeah, yeah, very cool. So, okay, so what I guess is what is motivating you in this field? Why, especially neurotech?
Yeah, so I was fortunate to have an experience, kind of two key experiences that led me down this road. The first was in high school. I took a systems physiology class, which was, I thought, in retrospect, pretty exceptional to have that opportunity. I went to a public high school in New Jersey, and it was a one semester class where we got to cut up cats that were, they'd bought from some, Carolina Biological Supply, probably, which is right down the road from us here. And that class really piqued my interest in looking at physiology from a systems point of view, which I think is a key part of biomedical engineering. And then as an undergraduate at Boston University, I worked in a research lab during my junior summer with a guy named Herb Voigt on auditory neurophysiology. So that was kind of my real introduction to the nervous system. And that experience really piqued my interest in doing something in neuro, but it also made me recognize that I wanted to do something that was a little more applied. So he was studying how is sound represented and processed in the cochlear nucleus, which may have implications for cochlear implants and is really important to understand, but was a little bit too far removed from application for what I was interested in. And I was fortunate to be able to connect with both Tom Mortimer and Hunter Peckham at Case Western and see the work that they were doing. And it was like a, kind of, bolt of lightning to see that, because it really was, it was exactly the kind of level that I wanted to be working at. the systems level where there was a clear potential impact on people with neurological diseases or disorders or injuries. And so, again, I think it's illustrative of the impact that mentors can have on you along the way, and kind of really drive your career forward. And now, I think one of the things that's most important about what we do is the training and mentoring. That, absolutely critical that the research that we do is improving our understanding about neural stimulation, interfacing with the nervous system, and we have clear examples where we've had direct impact on clinical therapies as a result of that work. And in parallel, the training and mentoring of young people who are going to be the ones that are going to carry this field forward, I think, is also of critical importance and has become enormously satisfying. So one of the things that I didn't anticipate that I would love so much about this job of professor is staying in touch with trainees from 20 years ago and seeing what they're doing now and being able to kind of relish the success that they're having in their own careers. And that's really just enormously fun and satisfying, and something that I did not anticipate.
Wow. Yeah. I mean, it's kind of like planting a little lemon tree, and then they kind of grow into a big tree or something bearing fruit and everything, right? Um,
Yeah.
but yeah, so not necessarily only into academia, right? But also maybe making their own companies, spin-offs, and I mean, you're talking about turning some of that research into, applicable, therapeutics and everything like this, right? And is it just kind of a standards of practice, or is it also companies? And how do you make sure that happens? Because I've always heard that it doesn't happen. Like a lot of times, research just kind of it doesn't it only goes to the bench. It doesn't make it all the way to the bedside.
Yeah, so just to answer the first part of that, most of our trainees do go into the medical device industry. Some go into academia, but by far most into the medical device industry, because that's where a lot of the jobs are. And I can give you an example. So Michael Moffat was my second doctoral student of my career. This was when I was a faculty member at Case Western. And he now directs a very large research group research and development group at Boston Scientific Neuromodulation. And he, a few years ago, hired another guy named Tianhe Zhang, who was a doctoral student of mine at Duke. And so this is now two generations working together to advance neuromodulation therapies at Boston Scientific. And so that's just kind of an example of the impact that your mentees can go out into the world and have. The second part of your question is, how do you ensure the impact not just of those people but also of your research innovations? And I don't know that there's any uniform way to do this. I think there are many, many different routes that I've seen this done successfully. I think one of the common features of success is investing the time and energy relatively early in the development process to understand the clinical treatment of the disease or disorder that you're hoping to impact now. So what are the drugs? What are the devices? What's the physical therapy that's used? Really understanding clinically what's happening. Who are the physicians that treat those individuals? Getting to know them at your own institution, spending time with them so you really understand the clinical side of things. Also understanding the market forces. How are those? Who's getting paid to do what in that therapeutic process? Because for better or worse, and that's a a different argument. Healthcare in the United States is a for-profit business. And so if you're going to get involved in moving therapies into clinical practice, you're going to end up interfacing with a for-profit business. And you need to understand a little bit about how that's operating if you think you're going to be able to come in and be successful. I'm not necessarily saying that's only true if you want to start a business, but just to have your therapy adopted, if it's inconsistent with the financial model, even if it's really good therapy, it may not be adopted. So I think these are things that as biomedical engineers, when we're in the lab, doing an experiment, we're not thinking very much about. And I think it's, if your goal is to do that translational step, these are important things to be thinking about relatively early on, because you can adapt your development path so that it fits with that clinical practice and that the economics of that market, so that ultimately what we want is the patients to benefit from that innovation. And if your puzzle piece doesn't fit there, not only do you lose, but more importantly, the patients lose. They're not going to have the benefits of that therapy. So making your puzzle piece oriented in the right way so that it fits in ultimately so the patients can benefit.
Yeah, that's really cool. I mean, I like this idea. I mean, I haven't really followed it, being intentional in the idea stage, right? Because that's when things can change the easiest, the fastest, the stakes are the lowest. Then when you get to the prototype stage, that's 10 times more effort to change something. And then once you get like to the market or, the small, small run, that's it. But then when you get to mass market, it's like impossible and, billions of dollars. So definitely be intentional in the beginning there. But I also see that as like an excuse to, do committees and to never really get anything done, and just, always, just be thinking about the problem and I don't know, just being stuck in the idea phase. So how do you kind of balance those two things, like actually getting stuff done, actually getting things out to mass market without, for lack of a better word, mental masturbation?
Yeah, I think there's an engineering, I don't know who it's attributed to. How about Yogi Berra, right? Better is the enemy of good. And I think you do have to be cautious about when is the right time to move your innovation into clinical use, right? Whether it's that first feasibility test or beyond that, trying to get it into real clinical adoption. Because we can fall into a cycle where you're constantly improving, perfecting and improving, make it work better, and because it's not, you say, well, it's not quite ready because it's not perfect, so I'm going to keep working on it, keep developing it, and not push it out the door to really see if it's going to is your puzzle piece going to fit. And I think part of there are two dimensions of that. One is risk. That is, all these therapies have risk associated with them. So let's take a cuff electrode. Right? We could design a cuff electrode that in 5% of the animals in which that electrode was implanted during a preclinical study, there was some evidence of mechanically induced injury to the nerve. And might say, well, that's unacceptable, so we're going to go back and redesign it and do another study. And hey, we got it down to 4%, but we think that's still too high. And okay, we're going to redesign it. We're going to do it again. We got down to 3%. Okay, now it's okay. I would advocate pushing that out a little bit earlier to understand whether there's potential for clinical efficacy, that then you might go back and say, hey, it looks like it might actually work, and now we should invest the time and money to reduce the risk associated with it, rather than reduce the risk down to 1%, push it out into the clinic and realize, well, it's very safe, but it doesn't do what we thought it would do efficacy-wise, and so it doesn't have value. And I think as engineers, we need to be really need to balance how much do we de-risk something versus when do we push it out there and see if there's the potential for clinical efficacy. I think the most important part, in
Yeah.
my opinion, of that equation is knowing what those risks are and being able to describe them clearly to our clinical colleagues, to potential study participants, to the IRB, to the FDA, and then people can make a judgment as to whether they think it's now is the time. Or they might come back and say, you know what, Warren, 5% is too high. You need to go back to the lab and reduce that before we're willing to take this forward. But I think we should be cautious internally about trying to push it down to 1% because that may not really be necessary.
Yeah, definitely. And I guess with the risk tolerance and cost benefit for, providing help for people at the cost of potentially side effects, would you say that that's one of the biggest challenges in your work?
What is the biggest challenge?
Basically cost benefit analysis, and I mean, potentially having people's lives at stake.
I think that I wouldn't. I mean, certainly the risk profile of everything we do is front and center, but I would not say that that's the greatest challenge. I think, for example, um, inadequate, or I wouldn't say inadequate, but animal models that are not entirely replicative of the clinical situation that we're trying to address is a bigger challenge. So I would say the efficacy side is more of a challenge than the side effect or risk side. And that, again, is, in my view, another reason to push therapies when advisable early into human. Because if your animal model only represents part of the disease process, then you can't really use that animal model to assess efficacy entirely. Push that over to human, for example, by doing intraoperative experiments or using minimally invasive approaches or temporary devices to understand the potential therapeutic impact in human early. Because if it's not going to work, you want to move on and do something else. And if it is going to work, that amplifies that we should go back and really continue to push on that idea in the preclinical arena to really get it right and then get it into human permanently.
Uh-oh, I hear another controversial statement coming up. Are you about to say that rat models are invalid?
That's not what I said, and please don't put that message out there at all. I said these animal
Okay.
models, all models, human and animal, have limitations. So, for example, we use a 6-hydroxydopamine lesion rat model of Parkinsonism, and to see the symptoms of Parkinsonism in that rat takes between 7 and 14 days after lesion. The dopamine neurons die as a result of administration of 6-hydroxydopamine. In humans with Parkinson's disease, that progresses over years before we see the motor symptoms. And so this is the rat is a model of Parkinsonism. It is not Parkinson's disease. That then has limitations associated with it. We still learn a tremendous amount by studying that model. It means that when you can, you want to also do the similar studies in humans who have had that disease develop over the course of years instead of days to weeks.
Okay. Yeah, it's something I've heard, that all models are wrong, but some models are useful. Obviously, it can't be one-to-one, but you can approach that, I guess. Yeah, I was getting worried there for a second. Yeah. Well, I want to be, Dr. Grill, I want to be very respectful of your time. Is there anything that we didn't talk about that you wanted to mention?
No, I don't think so. This has been great. I really appreciate you taking the time to learn a little bit more about what we're doing.
Yeah, maybe you might get an unexpected visitor coming up knocking on your door at Duke.
Yeah, that'd be great.
Excellent. Thank you.
Thanks. Have a good evening.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.